Hello everyone, my name is Joe Wilson. Today I'm going to talk about the determination of the gender, genes with hormones. Here is my agenda. Firstly, I will talk about sex organ, then move to chromosomes. Then I will talk about sex chromosome determine genetic sex. Then I will talk about sexual differentiation occurs early in development. Finally, we will talk about the hormone part, the female and male embryonic development. Now, let me begin our introduction with the sex organ. The male and female sex organs consist of three sets of structures: the gonads, the internal genitalia, and the external genitalia. Gonads are the organs that produce gametes, the eggs and sperms that unite to form new individuals. The male gonads are the testes, which produce sperm. The female gonads are the ovaries, which produce eggs or ova. The undifferentiated gonad cells destined to produce eggs and sperms are called germ cells. The internal genitalia consists of accessory glands and ducts that connect the gonads with the outside environment. The external genitalia includes all external reproductive structures. Sexual development is programmed in the human germ. Each nuclear cell of the body except the egg and sperms contain 46 chromosomes. This set of chromosomes is called the diploid number because the chromosome occurs in pairs. 22 match or homologous pairs of autosomes place one pair of sex chromosomes. The 22 pair of autosome chromosomes in our cells direct development of the human body form and of variable characteristics such as hair color and blood type. The two sex chromosomes, designated as either X or Y, contain genes that direct development of the internal and external sex organs. The X chromosome is larger than the Y chromosome, and includes many genes that are missing from the Y chromosome. Egg and sperm are haploid cells with 23 chromosomes, one from each of the 22 matched pairs plus one sex chromosome. When egg and sperm unite, the resulting zygotes then contain a unique 46 chromosomes, with one chromosome of each pair coming from the mother and the other from the father. Now, let's move to the next part. Sex chromosome determine genetic sex. The sex chromosome a person inherits determine the genetic sex of that individual. Genetic females are XX. And genetic males are XY. Female inherit one X chromosome from each parent, while males inherit an Y chromosome from the father and an X chromosome from the mother. The Y chromosome is essential for development of the male reproductive organs. If sex chromosomes are abnormally distributed at fertilization, the presence or absence of a Y chromosome determines whether development proceeds along male or female lines. The presence of a Y chromosome means that the embryo will become male, even if the zygote also has multiple X chromosomes. In the absence of the Y chromosome, an embryo will develop into a female. The sex of an early embryo is difficult to determine because reproductive structures do not begin to differentiate until 7th week of the development. Before differentiation, the embryonic tissue are considered bipotential because they cannot morphogenetically identify as male or female. The bipotential gonad has an outer cortex and an inner medulla. Under the influence of the appropriate development signal, the medulla will develop into a testis. In the absence of that signal, the cortex will differentiate into ovarian tissue. The bipotential internal genitalium consists of two pairs of accessory ducts. The warfarin ducts derive from the embryonic kidney and the medullary ducts. A development proceeds along either male or female lines. One pair of ducts develops while the other degenerates. So what directs some signal cell then to become male and some others to become females? Sex differentiation depends on the presence or absence of the sex-determining region of the Y chromosomes or SRY gene, as shown here. In the presence of a functional SRY gene, the bipotential gonads develop into testes. In the absence of the SRY gene, which we will discuss later detailedly, and under the direction of multiple female-specific genes, the gonads develop into ovaries. Now, let us move into the hormone parts. I will divide them specifically here into male embryonic development and the female one. Now, first begin from the male embryonic development. Testicular development does not require male sex hormones such as testosterone. The developing embryo cannot secrete testosterone until after the gonad differentiates into testes. Once the testes differentiate, they begin to secrete three hormones that influence development of the male internal and external genitalia. Testicular cellular cell secrete glycoprotein anti-Muller hormone. Testicular intellectual cells secrete androgens, testosterone and its derivative DHT, dihydrotestosterone. Testosterone and DHT are the dominant steroid hormones in males. In the development of the fetus, anti-Muller hormones cause the embryonic Muller ducts to regress. Testosterone converts the warfarin ducts into male accessory structures such as epidermis, vas deferens, and the seminal vesicle. Later in the fetal development, testosterone controls migration of the testes from the abdomen into the scrotum or the scrotal sac. The remaining male sex characteristics such as differentiation of the external genitalia are controlled primarily by the DHT. Now let me discuss about the female embryonic development, and there the picture shows the two different phases here. In female embryos, which has no SRY gene, we discussed before, the context of the bipotential gonad develops into ovary tissue. In the absence of the testicular AMH, the Muller ducts develop into the upper portion of the vagina and the uterus and the fallopian tubes. Without testosterone, the warfarin ducts degenerate. Without DHT, the external genitalia take on female characteristics. Now this is the human hormone part. Here is my reference I used for presentation today. If you have any questions, please contact me. Thank you.